We take for granted the sight of green on our planet, but for billions of years, the land was essentially a barren, beige wasteland.
Right, just sun-baked rock and sand. No trees, no flowers, nothing.
Then, about 500 million years ago, came maybe the greatest evolutionary migration the world has ever seen.
The movement of life from the, stable, buoyant world of water onto the really challenging dry land.
It's a fundamental planetary shift.
It is. If you want to understand life on Earth today, you have to understand the emergence of plantae, or the embryophytes. They didn't just survive on land. They re-engineered the atmosphere, the geology, and the climate.
Our mission today is to deep dive into that very origin story. We're going to trace the lineage from the aquatic ancestors and chart the absolute minimum set of evolutionary innovations.
The derived traits.
Exactly, the derived traits that allowed these first pioneers to not only survive, but, thrive in a world that was actively hostile to life.
We're charting, what, the first 100 million years of plant evolution? This takes us from the earliest microscopic spores, which originated around 470 million years ago,
Right.
through the humble ground-hugging mosses, and then culminates in the revolutionary rise of the first tall forests, the seedless vascular plants, around 385 million years ago.
Okay, so we have to start at the beginning. Who were the ancestors? Plants didn't just spring fully formed onto the land.
No, not at all.
They share a close kinship with a specific group of green algae known as the charophytes. What is the evidence that links all land plants back to this specific group, like the filamentous pond algae and the clade Zygnomatophyceae?
Well, it comes down to sharing complex, expensive cellular machinery. Plants and all green algae share basic features. They're photosynthetic, multicellular, and use chlorophyll A and B.
Sure, that's the broad stuff.
But the charophytes share three specific traits only with land plants.
And what are those key characteristics?
Critically, the first is the structure of the cellular machinery that's responsible for building their cell walls. If you look at the plasma membrane, both plants and charophytes have these distinctive circular rings of proteins. These are the cellulose-synthesizing complexes.
Circular rings, why does that matter? Most algae synthesize cellulose too, right?
They do, but most algae have linear arrays of these proteins. The fact that plants and charophytes share this specific, highly organized circular formation suggests a shared origin for their cell wall synthesis process.
I see, so it's a very specific fingerprint. What's the second link?
The second is that in the plants that still retain motility like ferns, their flagellated sperm structure closely matches charophyte sperm structure.
And the third, you said it's maybe the most crucial.
It is, and it relieves to surviving extreme environmental stress.
The stress of drying out or desiccation. They were living near the edge of ponds, constantly exposed.
Exactly. The crucial polymer here is sporopollenin. When charophytes living near pond edges reproduced, this incredibly tough, chemically inert polymer coated the diploid zygote.
So it's like armor.
It ensured that when the pond shrank or dried up, the next generation was essentially armored against desiccation, just waiting for the water to return.
And when the descendants of these algae made the great move onto land permanently, they co-opted that technology.
That's it. Land plants took sporopollenin and used it to coat their spores. This meant their reproductive units could be launched high into the dry, open air and dispersed via wind, traveling great distances without suffering harm. It was the original desiccation shield.
So this is where the complexity truly explodes, because surviving on land wasn't just about spore protection. Plants now face gravity, scarce water, and unfiltered UV light.
A whole new set of problems.
So what are the four main defining evolutionary innovations that separate the entire kingdom of land plants, the embryophytes, from their algal ancestors?
We can trace the entire plant kingdom back to four essential derived traits. The first is a completely unique life cycle, alternation of generations, or AOG.
This cycle is often where people get lost, because it's fundamentally different from the life cycles of animals or fungi. Can we slow down and walk through the stages simply?
We must. Unlike animals, where only the gametes are haploid, AOG involves two distinct multicellular stages that alternate.
Two stages.
You start with the multicellular haploid organism, which is the gametophyte. Because it's haploid, it produces its gametes, eggs, and sperm via simple mitosis.
Okay, so gametes fuse, forming the diploid zygote, and that starts the second stage.
Correct. The diploid zygote undergoes mitosis to grow into the multicellular diploid organism, which we call the sporophyte.
Right.
Now, the sporophyte produces haploid spores, but it has to do so via meiosis, the reduction division. Those spores disperse, settle, and grow via mitosis into a new haploid gametophyte, completing the loop.
The key distinction is the existence of two complex multicellular stages, one diploid and one haploid.
That's the core of it. The second trait is what gives the entire plant kingdom its name, embryophytes,
Which refers to the
to the multicellular dependent embryos. This is parental care, plant style. Unlike the algal zygote, which is just shed, the diploid zygote is retained within the maternal tissue of the female gametophyte, housed in a structure called the archegonium.
That makes sense for protection, but if the baby plant is retained, how does the mother plant feed it?
That's where the specialization comes in. They develop placental transfer cells.
Wow.
These are specialized cells in the maternal tissue that have these elaborate wall ingrowths, which significantly increases the surface area for efficient active transport. They are essentially nutrient pumps.
Pumping sugars, amino acids.
All the necessary molecules directly to the developing embryo. This ensured its high rate of survival in that new, tough terrestrial environment.
Okay, so the third innovation we already touched on, but it bears repeating.
Yes, the third is having walled spores produced in sporangia. The sporangium is the multicellular organ on the sporophyte that functions as a spore factory.
Further protecting the spores and reinforcing that sporopollenin armor.
Exactly, for air dispersal.
And finally, trait number 4, which allows for directed growth against gravity.
That's the apical meristems.
Yeah.
These are localized regions of, well, perpetual cell division found at the tips of the roots and the shoots.
So they're always growing.
Always. Because land is patchy, water is down, light is up, the plant needs to be able to continuously elongate. This continuous growth, powered by the apical meristems, lets them actively explore the soil for water while simultaneously competing vertically for sunlight.
They also needed specialized structures just to manage basic survival, like breathing and waterproofing.
Absolutely. The necessity of surviving desiccation led to the development of the waxy cuticle covering the epidermis.
Your primary waterproofing layer.
Right. But if you're sealed in wax, you can't photosynthesize. So they evolved stomata, specialized pores that allow CO2 intake for photosynthesis, but can close tightly in hot, dry conditions.
It's that constant crucial balance between gas exchange and water loss prevention.
It is.
And before true roots evolved, how did these first plants get the nutrients they needed from the soil?
They needed help. They formed critical, ancient symbiotic partnerships called mycorrhiza with fungi. These fungi dramatically increase the surface area for the plant to absorb essential nutrients.
And we have evidence for this.
We see fossil evidence for spores dating back 470 million years, which suggests these partnerships were essential from the very beginning.
That brings us to the first group to diversify and thrive on land.
The ground huggers.
That takes us into the nonvascular bryophytes. This grouping includes the 3 extant phyla, the liverworts, the hornworts, and the mosses.
And these 3 lineages represent some of the earliest divergences in plant history.
Right. And while we group them informally as bryophytes, they do not form a single monophyletic group. They are distinct early branches.
Their strategy was fundamentally conservative. Stay small and stay wet. How is their life cycle defined, especially compared to the later vascular plants?
Their entire existence is defined by gametophyte dominance. The familiar stage you see, that lush green carpet of moss, that is the haploid gametophyte. This is the large, independent, and longer-lived stage. So if you see the sporophyte,
It's small, brief, and entirely dependent on the gametophyte for nutrition.
this constraint, their size, is directly tied to their lack of strong vascular tissue for structural support. And they don't have proper loops, right?
No, they don't. They use simple filaments called rhizoids just to anchor themselves to the substrate. Unlike true roots, rhizoids lack conducting cells. They don't play a primary role in absorbing water and minerals.
They still primarily absorb water through their surface cells.
They do. And their reproduction reveals just how tied they are to moisture.
I was going to ask about that.
It absolutely does.
Yeah.
Their male structures, the antheridia, produce flagellated sperm. These sperm have to literally swim through a film of water, whether it's dew or rainwater, to reach the egg retained in the female archegonium.
That need for liquid water fundamentally limits bryophytes to perpetually moist environments despite their armored spores.
That's the major constraint.
Let's visualize the dependent sporophyte attached to that dominant gametophyte.
Okay, so it's typically composed of 3 parts. A foot embedded deep in the gametophyte, a stalk called the seta that raises it up, and the capsule at the tip.
And that capsule is the sporangium.
Yes, where millions of spores are generated by meiosis. And there's an amazing mechanism for spore release in many moss capsules.
The peristome.
That's the peristome, a ring of tooth-like structures around the mouth of the capsule. These teeth are sensitive to humidity.
So they react to the weather.
They do. They curl inward and close when the air is damp, trapping the spores, and then unfurl outward when the air is dry, slowly flicking millions of spores into the air when conditions are optimal for wind dispersal.
Mosses might be small and, primitive, but they have a massive ecological footprint. They stabilize ecosystems.
They certainly do. Take a species like Polytrichum, which grows in sandy areas. Research shows it plays a vital role in retaining nitrogen, drastically reducing the amount lost from the soil via leaching.
But the ultimate heavyweight is sphagnum, or peat moss.
Absolutely. Sphagnum is an incredible biological chemist. It releases phenolic compounds that inhibit bacterial decay, and it actively acidifies the water, creating a low pH, low oxygen environment.
So that combination almost completely shuts down decomposition.
It does, which explains why these deposits are such incredible preservatives.
Like the Tulland man.
Precisely. The Tulland man, a human body preserved for millennia in a Danish bog. The detail is stunning because the decomposition process was basically halted by the sphagnum-driven chemistry.
And the sheer scale of the carbon storage here is staggering.
It's a critical global reservoir. Peatlands cover only about 3% of the Earth's surface, yet they store an estimated 500 billion tons of organic carbon.
A third of the world's soil carbon.
It's a vast storehouse,
Right.
which of course ties directly into modern climate concerns.
Right.
If climate warming or human disturbance causes these peat bogs to dry out, decomposition resumes, releasing that massive store of CO2 back into the atmosphere. It's a powerful, positive feedback loop.
And some theories suggest these early plants actually did the opposite in the past.
Yes, one theory suggests the expansion of early non-vascular plants actually contributed to global cooling and glaciation during the Ordovician period by accelerating rock weathering, which pulls CO2 from the atmosphere.
It's amazing that these early life forms could fundamentally change the global thermostat. But now we move to the next great leap in plant history, escaping the ground-hugging life by inventing height.
This takes us to the rise of seedless vascular plants, appearing around 425 million years ago. This innovation was driven by the intense competition for light and the desire to disperse spores farther.
This means they completely flipped their life cycle strategy, right?
They did. We see the shift to sporophyte dominance. The diploid sporophyte is now the large, complex, independent, and dominant generation. The leafy fern you see is the sporophyte.
And the game to fight.
Greatly reduced, tiny, and often grows hidden just beneath the soil surface.
And this height was made possible by the complex vascular tissue system. We're talking about xylem and phloem.
Yes. Xylem conducts water and minerals up from the roots. Its functional cells are the tracheids, which are tube-shaped cells, dead at maturity. Crucially, their cell walls are structurally reinforced by the polymer lignin.
And phloem.
Phloem, in contrast, distributes sugars and organic products throughout the plant, and those conducting cells remain alive.
Is it fair to say that lignin, above all else, is the single biggest difference maker that created the modern forest?
I would argue yes. Lignin is incredibly tough and durable. It provides the rigid structural support needed to defy gravity and grow tens or even hundreds of feet tall.
Without it, you're stuck in the moss carpet.
With it, you get the first trees capable of accessing sunlight and outcompeting every other life form on the planet at that time.
Beyond height, vascular plants develop two other major components: true roots and complex leaves.
True roots evolved as specialized organs for anchoring and absorption, a huge upgrade from rhizoids. And we see two major evolutionary pathways for leaves.
What are those distinctions?
First, you have microphylls. These are small, spine-shaped leaves supported by just a single strand of vascular tissue. They appeared around 410 million years ago and are found only in the lycophytes.
And the second type?
Second, you have megaphylls. These appeared later, around 370 million years ago, and feature a highly branched vascular system enabling much greater photosynthetic efficiency. They're characteristic of almost all other vascular plants.
Reproductive structures evolved as well, with the development of sporophylls-modified leaves that bear sporangia.
We see this in ferns as clusters of sporangia called sori on the underside of the leaf, and this innovation paved the way for reproductive specialization. Most seedless vascular plants are homosporous.
Meaning one type of spore.
Right, one type of spore that develops into a bisexual gametophyte. but all seed plants and some seedless vascular groups are heterosporous.
Heterospore is the precursor to seed evolution.
Absolutely. Heterosporous plants produce two types of spores: large megaspores, which develop into female gamophytes, and smaller microspores, which develop into male gamophytes.
This specialization meant the separation of sexes.
A critical step toward the reproductive success of seed plants.
Today, the two major groups of seedless vascular plants are the lycophytes and the manilophytes.
The lycophytes we see today are small plants, but their ancestors were the dominant giant trees of the Carboniferous period. The manilophytes, particularly the ferns, are the most widespread group today.
And they're actually more closely related to seed plants than they are to lycophytes, sharing those highly branched megaphylls.
That's right.
The peak of this era was the Carboniferous period, starting roughly 385 million years ago, when these plants formed the first global forests.
This is a world of towering lycophyte trees, giant horsetails, and massive ferns. Their impact was, well, it was catastrophic for the atmosphere.
How so?
The sheer volume of their growth accelerated the processes of rock weathering, which pulls CO2 out of the atmosphere. This led to a huge drop in global atmospheric CO2 levels.
And a drop in CO2 equals global cooling.
Precisely. This biological activity contributed to significant global cooling and widespread glaciation. Then, as these forests died in the stagnant, oxygen-poor swamps of the Carboniferous, their lignified bodies failed to fully decay.
Leaving behind all that organic material.
These undecayed remains formed vast, thick layers of peat. And under immense heat and pressure, that peat transformed into what we now know as coal.
The fossil fuel age is literally built on the bodies of the first forests.
The ultimate legacy of the seedless vascular plants.
So if we look back at the entire journey of this deep dive, the three major evolutionary milestones were: 1, the crucial genetic origin from charophyte green algae.
Right.
2, the suite of adaptations for desiccation resistance, cuticle, stomata, and spore pollenin.
The whole survival toolkit.
And 3, the invention of structural support and plumbing with vascular tissue, especially the polymer lignin, allowing plants to conquer gravity and dominate the land.
The profound connection here is that plant evolution fundamentally re-engineered the planet. The shift from water to land wasn't just a biological success story. It permanently altered the planet's atmospheric composition and geological cycles. Life changed the Earth itself.
Which brings us back to that legacy you just mentioned, the Carboniferous coal.
Consider the incredible irony of this history. The fossilized remains of those first forests. The coal helped pull massive amounts of carbon out of the atmosphere during the Carboniferous period, causing global cooling.
Right.
Today, we burn that stored carbon, returning it to the modern atmosphere and contributing directly to modern global warming. It's a planetary-scale feedback loop that connects ancient life directly to our current crisis.
A compelling story of biology, geology, and climate all wrapped into one. Thank you for joining us as we explore the incredible colonization of land by plants.
We hope this deep dive leaves you with a profound new appreciation for the green world around you.